Dear Professor, thank you
Yes.
for your presentation. Now we have questions from our participants.
Sure.
The first one is, because mutation rate is fast, how effective RNA vaccines would be? Wouldn't they become ineffective after a while since they usually code for a certain part of the virus?
Yeah, that's an excellent question. So let's talk about mutation frequencies so we're talking about RNA viruses here. So DNA viruses can take advantage of the cellular repair mechanisms, the DNA repair mechanisms of a cell, but RNA viruses really can't. HIV, in and of itself, has the reverse transcriptase has a very high error rate. So for every time you get a strand synthesized, you have wind up with like 3 or 4 mutations per strand. It's a very high mutation rate. So you get swarms of variants made very quickly with HIV. You do with other RNA viruses as well. The coronaviruses are a little bit interesting. They're very interesting. They're different in a way, in that they have. I won't get into biochemical details, but they have what I would call a quasi repair mechanism. So that because of the way the virus replicates, there is some error correction in the thing. So the mutation rate would be the same for the actually strand synthesis, but in terms of it being, it can be repaired to a degree. So there's less, theoretically less variation. So now the question comes, if you make let's say you make a spike protein that covers one coronavirus, or you make an HIV spike protein that covers one type of HIV, and you go out and you use it as a vaccine. Well, these viruses evolve, so you wind up with resistant variants. In fact, the vaccine can help select for resistant variants in these cases if it's neutralization based, because so far as I know, neutralizing antibodies are escaped in all situations for all viruses that I know anything about. You might wind up having to do with SARS-CoV-2. We don't know this for sure. I mean, there's a lot of talk about the variants and whatnot. We don't really know for sure how much faster they spread. They look like some of them might. Looks like some might be more pathogenic. We're just in the middle of the science on it, okay? You see it on the news all the time as if it were a fact. But if you look at the science underneath, these are things you just got to get worked out. But worst case scenario, let's say we've got a variant that pops up that spreads and it's more pathogenic, et cetera. Particularly with the RNA technology, but also with the adenovirus technology, it's pretty straightforward to just make a new vaccine that has now the sequences of the variant viruses in it, and use that. In essence, that's what we do with influenza vaccine every year. The variation is not only by that's an RNA virus also, but it has viral genomic segments. So you have regular mutations, then you have genomics reassortment. And so every year we're making reassortment viruses, based on what's circulating for the influenza vaccine. So you wind up with a cat and mouse game, okay, which works. I think it's going to be, my hunch is, I could be wrong, it's going to be better for SARS-CoV-2 because I think there's less variation in it than there is for flu and certainly for HIV. For HIV, it's going to be a big problem, a really big problem with this. And this confronts everything where we're trying to make an antibody-based vaccine that was a neutralization. I'll give you an example. I mentioned my colleague Mo Sajati here at the Institute. Mo has isolated right now a monoclonal antibody out of one of his patients that has the greatest neutralization breadth of any known monoclonal antibody for HIV, okay? And this is going to take viruses from all over the world and doing it. And this Gates Foundation is supporting the development of this thing. But if you look at the viruses in the person he got this antibody out of, that person has a lot of resistant viruses, okay? So in the person where they've had this antibody and these viruses for 30 years now, this is a long-term survivor, this person who has it, you see escape variants. So this is going to be a problem we have to deal with one way or the other. Either live with it, learn how to live with it, or develop a vaccine strategy that avoids it. And there's a lot of interest in doing that sort of thing now. Okay.
Thank you. The other one is protected sex guaranteed to be safe? Can we say that sexual protection is enough to prevent HIV spread?
Nothing's perfect. As a rule, yes, protected sex is safe. That doesn't mean that that it's perfect. Okay, it's much safer than unprotected sex. The other thing, if a person knows that they're going to engage in high risk behavior and they're willing to do that, they can go on standard antiretroviral drug therapy, which a healthy person can take just fine. In fact, some of these now are injectable that will last for several months. And just having those drugs there will protect them almost completely. That's actually the safest way is if you're infected is to have protected sex or have protected sex along with the what we call PrEP, antiviral therapy for that. HIV transmission, the truth of it is HIV, if you do PrEP, if you do testing and contact tracing, et cetera, in theory, the epidemic or the pandemic can be stopped without a vaccine. But it's an expensive, difficult thing to do, hard logistically to do around the world, that sort of thing. And so it's thought that a vaccine would still be the best way to do it long term. So that's why we all keep after it. So.
Thank you. Next one is any long-term concern about this pandemic, including vaccines and how the behavior of people in global influencing the trajectory of the pandemic itself?
Yeah, that's a great question. I'm not an epidemiologist, but I've lived next door to them here in the Institute for years and influenced at least how they think. So it's a fantastic question. I can just see right now how you talk about changing risk-taking behavior. One of the consequences of the pandemic this year is interesting and sort of predictable if you think about it. Is at least in the U.S. And I think worldwide, we essentially have no flu. Because one here, at least in most of the country, people are being careful wearing masks, et cetera, and social distancing. Well, that also prevents flu transmission. Okay. The other thing is that there's north-south transmission of flu, depending on season. And all air travel essentially has been stopped north to south across the world. I mean, there's some, but not like it was. So we really have no flu outbreak as a cause of this. So I've always wondered, in the case of the U.S., we have anywhere between 50 and 80,000 deaths a year from flu that we don't, we think about it medically, we worry about it, but we go on about our lives. Okay. Now maybe by just wearing a mask at certain times of the year, for example, like Asian countries do, we can stop that. So maybe these kinds of good things will come out of it. The question is much larger than that, and I don't have any good answer for it because you get into things like vaccine hesitancy and vaccine distribution. Europe and the U.S. have and China have made the vaccines. Well, how do you get them to the other parts of the world? They need to get there as fast. They need to get there fast. Just logistically, and those kinds of things, are important too. So I just gave you a non-answer to a very good question. We'll see you next year.
Thank you. The next one is some people taking the vaccines are being infected with new types of coronavirus, and they are spreading it and getting infected themselves. So how can we explain this, and how can we pursue community to trust the vaccine since they are not seeing the results?
Okay, so you're talking about the infections in people who've been vaccinated. And thus far, most of the evidence for the lack of efficacy is from the AstraZeneca vaccine in South Africa, okay? These are very small numbers of instances that I don't know any scientific details about. These are the example. These are things that are getting reported in the literature before it really appears in the scientific in the press, before it gets published in the scientific literature. I haven't seen the data, so I can't give you a scientific opinion about it. Okay. What I do know is that most of the concern about the variants are based on in vitro methods. For example, you take vaccine sera for the Moderna vaccine, and you take one of the variants. You can show that that variant is maybe 30-fold, maybe even 100-fold less susceptible to neutralization in a test tube, okay, or in tissue culture. That doesn't translate to that much reduction in protection in vivo. Okay, it's very unlikely to. These are in vitro things. These are concerns. Okay, these are things that scientists work on, and we really would be better off working on them without every night a new variant popping up on the news before we've been able to figure it out. But that's life. So I don't know at this point how much this is going to attenuate the efficacy of the vaccines. Let's say some variants pop up that do. Well, the good news is, again, as I said, with these platforms, particularly the messenger RNA platforms, but also the adenovirus platform, it is, from a manufacturing standpoint, it's relatively straightforward. Not easy, but relatively straightforward now. We know how to make these things. To make a new variant, you make a gene, which takes you a day to synthesize. It takes you another day or two to put it in the vector, and then you manufacture it, okay? You're not making proteins and all this kind of stuff. So we can engage the cat and mouse chase. We're going to be a better cat because we have these new platforms to go after variants. How that turns in a year from now, I don't know. Okay, it just depends on the science. But I'd rather be in the situation where you're in right now with vaccines than not.
Thank you. Next one is there any cross-productivity or immunity against the prevalent variants of SARS-CoV-2?
Well, yeah. In fact, these vaccines protect overwhelmingly against the existing variants. You see some new ones out there, but even the new ones, say for the one case that I know about, and it's anecdotally from South Africa, they protect across there. There is some thought. SARS-CoV-2 is what we call a beta coronavirus. There are circulating seasonal coronaviruses. They don't cause death, but they cause what we would call the common cold. There may be some cross protection even against some of those. We don't know. There's a lot of interest now in trying to develop a vaccine that protects against not only against SARS-CoV-2 but against multiple coronaviruses. It's a good question, but I don't know. We'll see how it turns out scientifically over the next year or two.
Thank you. Next one is for someone that has high viral load, such as healthcare workers, would vaccination be more problematic compared to others in terms of side effects? If so, how can we solve this?
Yeah, typically, you would not vaccinate somebody while they have an ongoing infection with SARS-CoV-2. And the reason you wouldn't do that, there are several reasons. First of all, you're starting to make your own antibody in response to the virus. And if you have antibodies, antibodies can reduce the immunogenicity of a vaccine if you have the pre-existing antibodies there. The other thing is that the bad outcomes in SARS-CoV-2 are due to hyperinflammation. And you wouldn't put a vaccine, particularly an RNA vaccine, which actually gives you some inflammation. You wouldn't give that to somebody who has some risk of having a bad hyperinflammation and maybe, having a worse disease outcome. So what they're doing, I know what we're doing here in our hospital. In fact, my colleague Mosi Jadi, if you look, just this last week, published a paper on antibody responses in vaccine responses in healthcare workers who had been previously infected. Now they didn't have viral loads; they cleared the virus, and you get much better responses to the vaccine. There was another paper from another friend, another colleague, is in New England Journal saying basically the same thing. So at that point, you wouldn't vaccinate somebody who is in the middle of their infection. You treat the disease. You let it settle down before you give them the vaccine. And that's usually, a few months after the infection, things have settled down.
Thank you. Next one is, we know there is a serious debate about whether viruses are alive or inanimate. What's your opinion and why?
Yeah, this is where I go back to my structural biology background. We don't worry about those kinds of things. I'm more comfortable with a diffraction pattern from an X-ray than I am with that question. I don't know. It's a fun debate, but I let other people have it.
Okay, thank you. So the last question is, you said that viruses only bring bad news, but as far as I know, bacteriophages are the ones which can save us from antibiotic resistance. What do you think about it?
Yeah, that's great. In fact, I was hoping somebody would bring that question up. Okay, so bacteriophages. If you go back and look historically, bacteriophages were discovered by a Frenchman named de Herelle back in the early part of the 20th century. And when they discovered that they would lyse bacteria, they tried to use them as phage therapy. There were no antibiotics at the time. And if you look at the papers from back in those times, yeah, there were some examples of where apparently they cleared the infection. The problem with bacteriophage in putting them in a person is they're very immunogenic. And a lot of times you have antibodies pre-existing against common bacteriophages. If you've ever done phage neutralization, which we used to do a million years ago with antibodies, so it fell out of use, and when antibiotics came along, they quote unquote solved the problem. Well, with antibiotic resistance now, there's been a renewed interest in phage therapy. In fact, it was practiced in Georgia, and by Georgia I mean the former Soviet Republic, and also in the Soviet Union for a number of years. There's a good body of science now where they're looking into phage therapy with renewed interest to treat things like multiple antibiotic resistant bacteria. Okay, so I wouldn't count it out, but that's a great question, great point.
Dear Professor, thank you for your informative presentation and answers. And also, thank you for participating in our event. It was an honor for us to listen to you.
My honor as well. And hello to all my friends in Turkey. So.